All right. In this lecture, what we're going to do is we're going to put things together. Or, in other words, we're going to learn how researchers try to put things together. What I mean things is all our measurements and experiments and the things that we learn about the transcriptome, proteome, and the genome, etc. Okay? So how do we do this? We do this through a branch of science that's called systems biology. And that's what we're going to talk about. So basically, the goal of the systems biology is to put together all these knowledge about the proteins and genes and small molecules and RNAs that are in our cells and in the organism, for given organisms, into a picture that we can understand how they relate to each other. Okay? So what do I mean by that? So you can define a system, including a biological system, as interactions between components. Okay, so these are the interactions between components of a system makes up the system. So not only we need to know what the components are, and in our case, what are the components? DNA, RNA, protein, metabolites, microbes, right? So these are the components. So what we need to learn is how they interact with each other. And those interactions is going to tell us something about how the whole system works. Okay, so like I said, the components of us, basically a biological system, are the genes, RNA, proteins, etc. And the interactions can be various. You have already seen some interactions that are absolutely regulatory for an organism, right? Give me an interaction in the transcription regulation lecture that you think is very important for gene regulation. Interaction between two different components. All right, come on, guys. Let's wake up, wake up. What is it? Protein gene? What do you mean, gene? Excellent. Yeah, a transcription factor, a protein, binding to the regulatory regions of a gene and controlling the expression of that gene. Excellent interaction, right? If we knew all those interactions, that's if we knew all the transcription factors and their targets, it would really help us understand how the whole gene regulation works within the system, right? Give me another interaction. Post-transcriptional regulation. Not necessarily my lecture, but you should know anyway. Those are flip lectures. Give me an interaction there. Again, a regulatory interaction between two different types of components. Think along the transcription factor lines, but in this case, we're talking about post-transcriptional regulation. At some level, that's right. It's an interaction. But what I was thinking is very similar to how transcription factors control gene regulation through transcription, how microRNAs control gene expression, right, through binding to the mRNAs and controlling if they're going to be translated or not, or if they're going to be degraded or not. Right? That's another regulatory interaction between two different types of components, in this case microRNAs and mRNAs. Right? So these are the types of interactions that we're talking about. What's another transcriptional regulatory interaction between two proteins in bacteria that is important for regulation? Now we're talking about protein-protein interactions. In bacteria, prokaryotic transcription. Remember, the final is cumulative, so we're going to revisit back some of these concepts in the context of the larger course, but you should remember what were the critical steps in bacterial transcription initiation. Excellent, perfect example. What that is that the physical interaction between the sigma factor and the RNA polymerase was the interaction that was required for the transcription initiation, right? Again, here is an example of a protein-protein interaction that's very critical for you to understand that will tell you something about the system, in this case the bacteria, right? Perfect. So the interactions in this case could be protein to gene, that means protein binding and controlling gene expression, protein to RNA, it could be argonaute to microRNA, for example, protein to protein, protein to metabolite, for example, the glycolysis enzymes controlling the metabolism of glucose, right? So these are the interactions that we're interested in. And we have, throughout this course, this semester, you have learned several techniques. And some of these techniques that you learned will tell you something about these interactions, right? For example, for the interactions between transcription factors binding to gene regulatory regions, you've learned that ChIP-seq is a technique that will allow you to map where a transcription factor binds in the whole genome. So it's going to tell you all its target sites. It's going to tell you all its target genes for a given transcription factor. RIP-seq you have not learned, but it's very similar to ChIP-seq. Instead of immunoprecipitating chromatin, that's where ChIP gets its name from, right? It's ChIP, chromatin immunoprecipitation. In RIP, what you immunoprecipitate is RNA. So it's RNA immunoprecipitation followed by sequencing. In this method, what it allows you to do is it allows you to find out which RNAs a given RNA binding protein binds to. Okay, so that would be post-transcriptional regulation. And we're going to learn today about yeast-2 hybrid for measuring protein-protein interactions. All right. So in systems biology, we analyze these interactions as a network. So we talk about a network of interactions. And a network analysis tells us something about biological systems. So let's talk about what we mean by biological network. So a network is made up of nodes. So these are the nodes in a network and edges that connect them. So examples of networks, for example, in a social network, right, Facebook or whatever you guys are using these days, the node is going to be you. You're one of the nodes. And your social interaction, so your friending somebody else, is going to be the edge, right? So that's the interaction to another node, which could be your neighbor, your mom, probably not, your friend, etc. So a network of highway network, right, would connect cities. So in this case, the New York City would be a node. It will have a highway connection to Chicago. And then another network could be the genes. So in this case, the nodes could be genes, like a transcription factor. And the edge would be that transcription factor activating a target gene would be another node. Okay, so these are the type of edge and node interactions that you can measure. All right, so another term in networks is that for the hubs that have very many a high number of connections to another. For a given node that has a very high number of connections to other nodes, we call these nodes hubs, right? Just like that JFK is a hub for a bunch of airlines, right? Because a lot of the connections go through JFK. And the degree of connections means for a given node, how many edges does it have? So this node has 1, 2, 3, 4, 5, 6 degree of connections. Okay. All right. So here's a couple of type of networks. There are different networks with different properties. There are two different types of networks that I want to show you now and give you a couple of examples. So in one, you can have a random network. So what that means is that in a random network, contains nodes with randomly placed connections. What that means is for each given node, it has very few connections and they are randomly placed to their neighbors. So one example of a random network would be the highway system in the US. So each city would be connected to another with very few edges, very few highways, right? So this is going to be I-95, I-80, or whatever. So, and each node, so each city will be connected to its neighbor. And here is another type of network, which is called a scale-free network. So this would be like a what do you call it? Flights, right? So the airplane network across the U.S. would fit to this example, because scale-free networks contain hubs, which are shown here in red, with very high number of links. So a hub example, for example, would be JFK, Chicago, or Texas, Houston, or whatever, right? So these are the cities which have a lot of flights that are coming out of. So they're the hubs, which has a lot of edges, right? So they have a high degree of connectivity, right? And there are cities like Kansas, Wichita, or Lawrence, or whatever, right? All of those flyover states with cities that have one or two flights. They would be the nodes here will have fewer connections, right? Does that make sense? Okay. So how do we actually compare these type of networks? We compare these networks based on how each node is connected. Okay, so what we're doing here is, what we're plotting here is the distribution of node linkages. What that means is I'm going to this random network and looking at one node and counting how many edges it has, right? It has 3 edges. And I look at this node, it has 2 edges. This one has 4 edges. This one has 3 edges. This one has 1 edges, etc., right? Then I'm counting how many nodes have 0 edges, then I plot it here. How many nodes have 1 edges? I plot it here. How many nodes have 2 edges, 3 edges, 4 edges, 5, 6, 7, 8? And in a random network, because the nodes have very few connections, you don't have hubs. Those are the nodes that have many, many connections. So if you then plot the distribution of number of connections each node has, right, you're going to have an average number of connection for a given node, and it's going to follow this distribution because you're going to have few nodes that have very few connections, and you're going to have few nodes that have a lot of connections, right? Does that make sense? Okay. In a scale-free network, you do exactly the same analysis, and you plot the number of connections each node has. Okay, you determine the number of connections each node has, and then you plot the number of nodes that have a given number of connections. So in this case, you have these cities with 1 or 2 flights coming, 1 or 2 connections. Then the vast majority of the cities in the U.S. are going to have flights to a few cities, right? So you're going to have a high number of cities, high number of nodes with very few connections. And then you're going to have cities that are bigger, right? Like Kansas City would have more, will be connected to more cities than Lawrence, Kansas, right? So you're going to have the bigger cities, but there are fewer of them with high number of connections. And here you're going to have your hubs, which are very few cities, which are very few number of nodes, but they have a very high number of edges, a very high number of connections. So what the distribution of that is going to follow this power law. And if you take a logarithm of that the same distribution on the x-axis and the y-axis, it's going to show this kind of distribution. So when you have a network and you basically do this analysis, if you see a distribution like this for the distribution of node linkages, you're going to say that this is probably a random network. And if you see a distribution like this, you're going to say this network has a bunch of hubs, and most of its nodes have few connections, so this is a scale-free network. Okay? Make sense? All right. Examples. So in biology, most of the networks are scale-free. That is, we have a few hubs and a bunch of other nodes that have fewer connections. And that's also true for a lot of networks that you might be aware of. For example, a social network is a scale-free network, right? Some of your friends have thousands of friends, and some of your friends have fewer friends, right? And then, for example, in Hollywood, if you take all the actors as the nodes and then put an edge between them, right? So if two actors are in the same movie, you put an edge between them, so that the edges would be the appearance in the same movie. What you're going to find is some actors like Samuel Jackson is going to appear with many, many people because he makes lots of movies. And then some other actor, I don't know, who hasn't been making a lot of movies? Chevy Chase? I don't know. Whoever, right? So whoever might be a little more selective in the number of movies he makes, then is going to have fewer edges. All right. So what does this mean in terms of biology, right? How is the property of the different types of networks actually affect their robustness? What I mean by robustness is how easy it's going to be for that network to collapse if there was an attack. Okay? So the random networks are these networks that, like the highway networks, it's going to be fractured by any attack to any of the nodes. And that's usually because to go from New York, for example, to Chicago, you're going to have to go through many of these nodes, right? And if I-80 is basically closed due to an accident, you're going to have a very hard time coming out of New York, right? So any breaking in the edges within this random network is going to have a detrimental effect for the rest of the network. Does that make sense? However, so you're going to have all these failed nodes. In a scale-free network, a random attack to any of the nodes or the edges are going to not matter very much, because what are the chances of having an airport closure in Lawrence affecting the flights out of JFK? It's very few. It's very little, right? Because most of these nodes in a scale-free network is going to be having fewer connections. So if you attack them, or if there's a closure, it's not going to affect very much. However, in a scale-free network, if a hub closes, right? So snowstorm in Chicago, that's going to have ripple effects for everybody else flying in the U.S. because it's a hub. So a problem within a hub is going to be then detrimental for the rest of the network. Does that make sense? All right. So, in summary, scale-free networks have a few hubs, but all the other nodes are weakly connected. And we know that a lot of biological networks are scale-free, and this actually explained some of the earlier observations that the biologists have made, but because they weren't thinking at a systems level, they didn't know how to explain this, but now we understand. Because this kind of scale-free network basically provides a quantitative explanation for a phenomenon well known to biologists, that is that biological organisms are unaffected by most random mutations, so you can delete or make mutations to a great number of genes in our body, and it's not going to show much of an effect. But mutation affecting any of the hubs are going to be lethal, right? Give me a hub gene in humans. That a mutation in that hub is going to be lethal for the organism. Give me a highly connected gene. P53. And your reasoning, because it's lethal, it probably has a lot of connections, and that's true. It has a lot of connections, so it's going to be lethal. All right. All right, so next what we're going to go through is we have seen ChIP-seq and other type of techniques that can analyze interactions. And today we're going to talk about a technique called yeast-2 hybrid that allows you to test if two proteins interact with each other or not. Okay, so right now you're then measuring protein-protein interactions. So why do we understand protein-protein interactions? Because you want to understand the network of physical interactions between all proteins in a cell that carry out the cellular functions encoded in the genome. For example, a lot of these molecular machineries that you have learned that are important for very important cellular processes are existing as protein complexes. We talked about transcription initiation complex. It has hundreds of proteins. We talked about mediator, right? It has many, many numbers of proteins. And you talked about other protein complexes. In addition to protein complexes, we talked about dynamic interactions that actually regulate the outcome of a cellular process, which one of your friends just said, the sigma factor interacting with RNA polymerase was one of these type of dynamic interactions, right? So how do we do this? We can use yeast-2 hybrid. Yeast-2 hybrid technique measures binary interactions. That is, basically, does the interaction occur or not? So what you do in this technique is you build two plasmids here and here. And what you do is you build these, so you clone a part of your gene that encodes for the protein that you want to test, right? So for example, if you are interested in all the protein interactions that one of your candidate proteins it makes, for example, so that would be your bait protein, right? And you would clone the DNA fragment encoding for that bait protein into this yeast plasmid. And what you do is you clone it in a way that fuses your target protein, right, the protein, your bait protein, to the DNA binding domain of a yeast transcription factor. In most cases, this is the GAL4 DNA binding domain. So when this plasmid, you put it into the yeast, this fused gene is expressed. It's going to produce a protein in which your bait protein, the protein that you're interested in, is fused to the DNA binding domain of the GAL4 transcription factor. Okay? So then you build a second plasmid. So in this plasmid, what you do is you put another protein, right? You're going to clone another protein in a way that when it's expressed, that protein, in this case, this is the fish, will be fused to the activation domain of a yeast transcription factor. In most cases, this is going to be GAL4 activation domain. Okay? So basically, you're going to build two plasmids that is going to, if you transform it into the same cell, you're going to have proteins that looks like this, which is your bait protein fused to the DNA binding domain, and this, which is your test protein that's fused to the activation domain. In yeast-2 hybrid, we usually don't look at just the interaction of the bait with one candidate, right? You can do that kind of analysis as well. If you suspect that your protein is interacting with another protein and if you want to test if it is physical interaction, you can do that. But if you're interested in all the protein interactions that your protein can make, what you need to do is then you need to make a library that is going to have protein 1, protein 2, protein 3, etc., however proteins that you can however many proteins that you clone into a library of the fish vector, right? Does that make sense? So you're going to have all different kinds of proteins that are cloned, right? So you're going to have a library of this fish vector. What do you do next? Then the principle behind Y2H that's yeast-2 hybrid is this. What you have is that you have a reporter gene, right? Just like how we analyze gene expression, reporter. And what you have here is you have reporter gene that is cloned downstream of a upstream activating sequence, which you know binds to Gal4 DNA binding domain. Okay? So what you have here is your upstream activating sequence that's binding to the DNA binding domain, and that also contains your bait protein, right? That's the protein that you're working on. And if your protein actually interacts with the fish protein, so that is one of the proteins in your library that you're screening for, what that does is it brings the activation domain of the Gal4 proximity to the promoter. So it recruits the activation domain of the Gal4 to the promoter of the reporter gene, right? And when the activation domain of the transcription factor is there, it's going to be able to activate the transcription of the reporter gene. In this case, that's the His gene. So it's going to then transcribe these mRNAs. So if this interaction occurs, then these yeast cells should be able to grow on plates that lack the histidine amino acid, right? Because you learned that in yeast auxotrophs, right, in yeast strains where they cannot grow in the absence of a given amino acid, you can actually complement that with the gene that encodes for the enzyme that makes that amino acid. So these cells are going to be able to grow in minus His media. Okay, I'm going to pause a second. This is really important. So if you have any questions, tell me about this. Just ask me now, okay? Does that make sense? This works, right? This works because transcription factors are modular. They have a DNA binding domain and an activation domain, and they can work independent from each other. So you take advantage of that, and you basically build this kind of little module here, which is that you fuse your protein of interest to a candidate, and if they physically interact, you bring the activation domain for expression. Question?
Why can't you use like GFP for the reporter gene?
You could use GFP for the reporter gene, but for a lot of yeast assays, these auxotrophs are really great to use because the outcome of the assay, you don't need a microscope. What do you need is basically a plate. If you see a colony growing, great, that gene is expressed. If you see a colony not growing, you know, okay, that gene is not expressed. So the detection is super simple. Any questions? Okay. All right, so basically in this assay, what you're going to have is you're going to have your bait gene, you're going to have your the fish cDNA library that is going to express all the proteins that are fused to the activation domain, and you have your reporter, which is going to express this his gene. All right, so if you build these plasmids so that you also have a bait library, that is, you can clone different proteins into the bait plasmid, right? Then what you can do is you can measure the interactions between protein A to protein A, protein A to protein B, protein A to protein C, etc., as well as protein B to protein A, protein B to protein B, protein B to C, etc., right? So what that means is that you're going to build these bait vectors, and you're going to have multiple vectors, right? And each of these vectors is going to contain protein A, B, C, and D, and you're going to build these fish vectors, right? And each of these fish vectors is also going to contain protein A, B, C, and D. And when you co-transform these two plasmids, what you would be looking for is you'll be looking for physical interaction between protein A and protein A. If you transform these two together, you would be measuring interaction between protein A to B, etc., right? And what you need to do then is you need to assay if the reporter is expressed or not. So for this one, what you're going to do is now you're going to transform your bait and fish into the yeast that are auxotroph for trip and leucine. Because you have these trip and leu genes that are in your plasmids, so for any yeast cell that contains these two will be able to grow in minus strip, minus leu plates, right? So if a cell contains these two plasmids, they should be able to grow. And that's kind of, I drew it as like a blue colony. So that's going to grow. And so you make these, so you have all of these, right? And then what you can do is you can replica plate this, so you make a copy of this plate, but this time, in addition to not having trip and leu, to ensure that these two plasmids are there, what you're going to do is you're going to make a plate that doesn't contain any histidine, right? So only those cells that express the hist gene are going to be able to grow on this plate. Then what you're going to do is you're going to look, okay, do I get a colony from A to A interaction experiment? No. So that means that protein A doesn't interact with protein A. Do I get a colony from A to B plasmid co-transfection? Co-transformation. Transformation, it's pretty much the same word. It's basically getting into a cell. The convention is that we use transformation for yeast and bacteria. It depends on the technique, but don't worry about it. What I mean is that getting the plasmids inside the cell. So if you see a colony, then for when you have both A and C, what you're going to say is that the protein A and protein C interacts. Does that make sense? All right. So now let's take this data and actually form a protein interaction network with it. It's actually very simple. What you're going to do is you're going to look at this. You're going to look at this data, and you're going to form edges between two proteins where you see an interaction. Okay. So you're looking. Let's look at A. Doesn't interact with itself, but it interacts with B. So I'm going to put an edge between A and a B. It interacts with C. So I'm going to put an edge here. It interacts with D, so I'm going to put an edge here. Then I'm going to look at B. B interacts with itself, right? So B interacts with A, so that interaction we already knew from looking at the other way, right? And then B also interacts with itself, so you put a little circle here, interacting with itself. So there's an edge between, that's connecting itself to itself. It does not interact with C, and it does not interact with D. And you go do the same thing with C. C interacts with A again, which we knew. And C interacts also with D, right? And then you go next in line, where D interacts with A, which we knew, and D interacts with C, which we knew from the other test. Make sense? All right. So what we did now is a protein interaction network that basically connected several nodes with edges. So what are the nodes in this network? Speak up. Yeah, proteins. And what are the edges? Physical interactions. This is physical interaction, right? Interactions can be different. It could be physical, it could be genetic, it could be regulatory. This is physical interaction, right? Protein binding to each other. All right. So here is an example in which this yeast-2 hybrid is done at a high throughput. So instead of just looking at 4 proteins, you can look at hundreds of proteins at a time. So in this case, in this example here, what I'm showing you are the yeast growth plates that have the histidine. So all the colonies that have the 2 plasmids should live here. And you see these tiny little dots, those are the colonies. And here is the plate that lacks the histidine. So only those pairs that interact are going to be able to grow on these. And so you can look at these colonies and find out which proteins are interacting. So in this case, what the bait was a PCF11, which is a component of pre-mRNA cleavage and polarization factor. And the prey was the 6,000 yeast proteins. So they tested this and said, which of these 6,000 yeast proteins actually interact with PCF11? So what they found is they found 11 colonies that can grow on the His minus media. So there were 11 interacting proteins. So they knew that 3 of them were previously known to be interacting. So kind of validating the assay is working, right? And then what they did is they repeated it for 6,000 other proteins. So for after doing it with PCF11, they did it for every single yeast protein. So what that means is that they basically measured 6,000 to 6,000 interactions, right? 6,000 to 6,000 one-to-one interactions. And that is 36 million interactions in total. And what they found is they found 957 interactions that connect about 1,004 proteins. So this was done quite a while ago, so in 2000, and it's been published. And when you do the network analysis, just like we did, right? So in this analysis, they just have more numbers instead of our 4 little toy example. So each of these little nodes are the proteins, and each of these edges are the physical interactions between proteins. So when you put the network like this, what you can see is that it's like a hairball, right? But what you also can see is that there is a node here that's very highly connected, right? And what kind of network does this resemble? Random or scale-free? Scale-free, that's right. So next what they did is that in the previous years, there was another study. And in this study, what the researchers had done is they took yeast strains and knocked out a single gene. So they made a library of yeast strains where each yeast strain had one gene knocked out, okay? And then they grew these yeast strains on media and looked which ones grow. The ones that grow, that knock out, not that important, right? The ones that don't grow tells you that the gene that was knocked out is essential. That means essential for growth, okay? So what they had basically is a phenotype measurement. So some genes knockout was lethal. Some genes showed knockout was slow growth, others non-lethal, and others unknown. These are not tested. Okay, so what they did is then they color-coded this network for each of the nodes, for them being either lethal or slow growth, etc., etc. Right, and what they could see is that they could see. Okay, so a lot of the genes are non-lethal, so you see lots of green ones, and some of them are lethal, they're red. And they asked, okay, we know that in scale-free networks, an attack to the hub is lethal, right? So they hypothesized that if a gene that's highly connected is knocked out, the chances of that gene knockout showing lethality or slow growth should be higher, right? If a gene is more connected, knocking out that gene is going to have a higher effect. Now they had this network, they can test that hypothesis, right? And they did that by making this plot. So what they did is they counted each gene and each node and said how many edges does that gene has, right? So what they have is in the y-axis you have the number of edges. And on the x-axis, you have number of edges. And on the y-axis, they said for that group of genes, for example, here, that has 5 edges, what percentage of those genes were essential, right? And they plotted that right here. For the group of genes that have 10 edges, what percentage of them are essential? And for a group of genes that have 15 edges, what percentage of them are essential? So what you can see here is that as the number of edges increase, the proportion of essential genes within that group increases, suggesting that is a positive correlation between how many edges a node has and the effect on the growth in yeast. Okay? So that means, in other words, more highly connected proteins, there are the hubs in the network, were actually found to be 3 times more likely to be essential, that is required for growth, than proteins with fewer number of edges. All right? All right. So this type of analysis have been also used to analyze human proteins. So this is the human protein network. In this case, 8,000 proteins have been tested against each other. So this is that many number of interactions have been tested, and they have found 2,800 interactions. So that was about 5 years after the yeast paper. And here what I'm showing you is just the part of the network, because the network is huge. You're not going to be able to see anything. But what you can appreciate is the researchers found that the type of observations they made for yeast network was actually very similar to the type of observations they made for the human network. That is, it looked scale-free. The essential genes basically were correlating with the number of interactions they have. And one example here is, for example, this is a gene this is a gene encoding for a protein that has a very high number of physical interactions, and that turns out to be this Ewing sarcoma-related protein, EWS gene, right? All right. So these are protein-protein interactions. So the edge is physical interaction. You can have protein-gene interactions. This could be regulatory interactions. For example, a transcription factor regulating the expression of a gene. Doesn't mean that the gene and the transcription factor are physically interacting. It means that the edge is that the knowledge of that transcription factor binding to the regulatory region of the gene and regulating the expression of that gene, right? And there are other interactions like this. One is called a genetic interaction. Some of you guys are taking genetics, so this should be pretty easy to you. But for others, in our genome, there are numbers of genes, right? There are 25,000 or so genes. So a genetic interaction can be in many different ways. For example, one way 2 genes can interact is what we call synthetic lethality. Okay? So what that is that if there is a combination of mutations in 2 or more genes that lead to cell death, whereas a mutation in only one of the genes does not. Okay? So you have a BRCA1 that's mutated, let's say. Actually, that's not a good example. What can I give you a good example? I don't remember. But what that means, I'll give you this example. So imagine the organism is like a airplane, okay? So the airplane has 4 engines. That imagine these are the 2 copies of gene A and 2 copies of gene B, the maternal and paternal, maternal and paternal. And both of these genes are important for a similar function, right? Let's say these copies are important for the function of this particular engine, etc., right? So if you have a mutation in one of these genes, what you're going to do is you're going to impair the function of one of these engines, right? This here. But then if you have a mutation in the other engine as well, now you're in trouble, right? Because if you make a mutation in only one of them, the airplane is still going to go, so the organism is going to live. But if you make a second mutation at another gene that's also important for the same process, then you're in trouble. That's basically what I mean by synthetic lethality. right? And in genetic terms, what that means is, what that refers to is what we call epistasis. That is the effect of one gene being dependent on another gene. That the phenotype that you see, right? So you knock out gene A, you don't see much by itself. You knock out gene B, you don't see much by itself. You knock out gene A and B together, now that's a problem, right? And these also occur in biological systems a lot because genes are redundant. What redundant means is that two genes might have similar roles on the same function, right? So the genomes are basically have two genes doing similar functions. So if you knock out one, you're still alive. If you knock out the other, you're still alive. But if you knock out both, then you're not alive. So this is kind of related to genetic robustness. That means if you have multiple genes doing similar functions, then you're going to be robust to mutations for individual genes. Any questions about this? Okay, so how can this be used to look at genetic interactions? This is typically done in yeast as well. This was pioneered in yeast because yeast is an awesome genetic model where you can do a whole bunch of stuff, in part because it's haploid. That means it only has one set. It can live as having one set of chromosomes. So what the researchers have done is they built yeast strains that have a yeast knockout library. That means a bunch of strains that have a knockout of a single gene. Okay, and yeast has actually two sexes, which are called mat alpha and mat A. So these are mating types, alpha and A, and you can actually have them mate, which forms a diploid organism. And through some genetic tricks that I'm not going to go through, so it's not important, what you can do is you can generate a bunch of progeny that has a progeny that contains the deletion of both genes that you initially made it. Okay? So it is basically a way in which you can create a strain that has the deletion for two genes instead of one. Okay, so you combining two knockouts into one strain. So then what you can do is you can say, all right, I know that this knockout grows. I know that this knockout grows. When I put them together, does it grow or not? And here's an example of that. What we're looking at is all of those little yeast strains that contain they are double mutant, so they contain two mutations, two knockouts that have not grown. Okay, so what that means is that if you have two genes when they're not growing because there are mutations in two different genes, those two genes genetically interact. Does that make sense? So that's the synthetic lethality interaction. That kind of analysis has been done for yeast. So what the researchers have done for every single gene knockout, they tested if it interacts with other genes, and they formed a network again. So in this network, what are the nodes? In genetic interaction network, what are the nodes? Genes. What are the edges? Okay, you guys need to articulate these things because when we ask in the midterm, you need to write it, right? What are the edges? Genetic interactions, in this case, synthetic lethality, right? If you wanted to be more specific. All right. So that's the network here. And what we're doing is we're zooming in certain little clusters. So these clusters are basically those genes that interact with each other more and not so much with each other. And now what these researchers have done is after they built the network, they color-coded those clusters for certain functions. based on how many of those genes in that cluster were known to have that function. Okay? So here's a cluster here. Here's a cluster of genes that interact with each other, and they knew that a bunch of them were involved in autophagy, so they basically color-coded them in red. And what you can see is that those genes with known functions, they tend to cluster together, right? Because they're doing similar functions, for example, here and here and here. And here are some examples, right? So can someone then tell me, how can you use this type of genetic network analysis to help assign or predict gene function for genes that you don't know what their functions are?
You can run an assay of the new gene against all of the genes that you know and then see where it fits in the network.
In the network, let's assume that you've done it for every gene. So you've done that already, and you have clusters in which you know that the genes within that cluster are enriched for a given function. What if you find a gene in the same cluster that have, for example, you found a gene here that is clustering with all these particular signaling genes, but you don't know what its function is. If you were a researcher, what would you do? What would your hypothesis would be for that gene? Because that's your observation. You observed that an unknown gene is clustering along with a bunch of genes you know are involved in a particular signaling pathway. What would your hypothesis be based on this observation? Come on. That's it's involved in signaling, and that's a very nice way to articulate. It's short, you just said it. That would be the hypothesis, that it is involved. It might be also involved in signaling. Then you can take that hypothesis and follow up with experiments and test if it's involved in signaling. So these kind of the network analysis help you predict gene function for unknown genes, right? That's pretty cool, actually, given the fact that I told you guys that half of the genes in any given genome, we still don't know what their function are, right? So it gives you a really nice tool to predict function. All right, so these kind of genetic interaction assays can be extended in humans as well, and these are usually done in dishes, right? Because you can't do experiments with humans. So basically what you can do is you can knock down a gene. In any given human cell, you can knock down a gene using RNAi, right? You guys learned how RNAi works. So you can use reagents to knock down a gene's expression using RNAi. And you can see a phenotype, let's say growth, how it affects growth. And now what you can do is you can do it for every single gene in the human genome, right? And then what you can do is you can do double knockdowns. It's a very similar idea, right? Instead of double mutants, you're using double knockdown. So what you can do is you can knock down 2 genes in the cell and see how that affects growth. And then you can take and phenotype that and convert it into a interaction matrix and then build interaction networks, genetic interaction networks for humans. So that's with RNAi. So now think about all our lectures in the past and give me another technology that you might be able to use to make genetic interaction networks in a very similar way as people have done in yeast. Instead of RNAi, what can you do to impair 2 genes at a time given in a human cell? CRISPR. So if you could make target RNAs to individual genes, right, you can transfect the cells with 2 different targets to 2 different genes, and that should allow you to be able to interrogate if those 2 genes actually interact or not. All right. You can have all kinds of fun with these type of experiments and networks, and you can test different types of interactions, right? And one of the interactions that people have looked at are co-expression interactions. So that says, okay, if a gene has the same expression profile upon a certain signal or time or something, right, with another gene, it's probably true that it's your hypothesis that those two genes are connected somehow. So they're co-expressed. That usually means they might be co-regulated. That is, for example, if there's a set of genes in the muscle cells that are all targeted by MyoD and they're all activated, right? What you might see is that those two genes that are regulated by MyoD will show a high correlation in their expression pattern. Does that make sense? So what you can do now is you can make a network in which the nodes will be the genes and the edges are going to be the correlation of expression, right? And these are called expression networks. So what can you do with that? Because you can measure expression quite easily. You can create all these networks, and you can do it in several animals. For example, here's an example of a network with co-expression connections done in humans that is basically compared to them the same type of interactions that are observed in chimpanzees. So these researchers were looking for hubs that were highly connected to humans but not connected in chimpanzees to try and get down to what makes humans and chimpanzees different in terms of molecular level, right? So how do you then calculate these edges, right? Edges, I said, are correlation of expression between two given genes. What kind of technique are you going to be able to use to be able to calculate expression co-expression correlation? What techniques can you use at a genome scale? Microarray. Give me another one. RNA-seq. I hear you guys murmuring. Just speak up. It'll help me out. All right. Excellent. Okay, so you can basically build networks for any type of interactions that you want to analyze. Basically, what you need is a technique that will allow you to measure the interactions and quantify it, right? And then it needs to be high throughput. That means you should be able to do it for many, many, many genes, so then you build a network. So how can you do this? For example, you can look for microbe correlations to predict microbial interactions. We talked about microbiomics, and that might allow you to look for microbe interactions. Let's say that you're working in a gastrointestinal disease lab, and you want to know which microbes are interacting with each other. What kind of experiment will allow you to build a network of microbe interactions? What would you do? How was the human microbiome experiment done? You can take a bunch of people, right? And then you can take gastrointestinal samples. You're going to sequence to find out all of the bacteria in the guts, right, of these different people. Let's say a bunch of healthy people and a bunch of sick people. And then what you can look for, just like gene co-expression experiment, what you can do is you can look for microbe co-presence, right? So you can measure that the edge should be quantitative, right? So you can put a number to how many times, for example, you see two microbes present in the same sample. And that could be the edge between the two microbes, right? And taking that information, then you can build a network in which the nodes are going to be the microbes and the edges are going to be what are the how many times did these two microbes actually were present in the samples that I looked at, right? Make sense? All right. Along the similar lines, you can look at microbe-metabolome interactions. What you would do is for each sample, you can determine how many microbes are there. I mean, you can determine which microbes are there, and then you can also determine which metabolites are there, right? And if you do this many times, you might be able to correlate the presence of a metabolite to the presence of a microbe, right? So you could do protein metabolome interactions and transcription factor, transcriptome interactions, etc., etc. All right, here's an example by which these researchers use the ideas behind these kind of experiments to actually attack a clinical problem. So, as you know, that in hospitals there have been a lot of antibiotic resistance bacteria, and these are horrible because if they basically get into the ICU or some patients that are have lowered immune reactions, these bacteria can be very deadly. And in fact, this is one of the problems that a lot of hospitals are facing. And so one of these type of bacteria that have a high antibiotic resistance is called Clostridium difficile, right? And so basically it increases the morbidity and mortality of hospitalized patients. So these researchers said, okay, when these patients are going into the hospitals, because we do not want them to get infected, they put them under heavy antibiotic treatment, right? And what it does is it clears the microbiome of that patient. So that patient doesn't have any bacteria anymore, right? And if that patient who doesn't have any bacteria actually gets this antibiotic-resistant bacteria, what happens is that bacteria immediately grows in the patient and makes that patient sick, and that person dies, right? They said, is it possible then that is actually one of the reasons why this is more dangerous. Furthermore, they made another observation in that some patients were more susceptible to this infection and dying than other patients. And they said, okay, is it possible that some patients have some remaining bacteria, right, that actually competes with this Clostridium difficile and actually helps that patient to fight it, right? So what they did is they said, okay, let's try this in mice. So what they did is they treated the mice with different antibiotics. So that results in distinct microbiota changes. That means different antibiotics are basically killing a certain family of bacteria, right? So they basically affected microbiome of these mice in different ways. And then what they measured is they looked at the varied susceptibility to Clostridium difficile. And what they did is then they used mathematical modeling to find out which of the remaining bacteria were most correlated with a high resistance to Clostridium difficile infection, right? And if they knew that bacteria, they said, okay, we have some candidate bacteria. Then what they did is they took that bacteria and gave it to mouse, right? And then infected the mouse with Clostridium difficile and see, okay, if we give the certain bacteria to mouse, did it actually confer resistance? And they found that in fact, upon administration, this bacteria, which makes this certain intestinal bacterium, enhances resistance to infection in a metabolite-dependent manner, right? So what you can see in this example is that you can actually look at interactions to be able to learn something about the biology and the biology of this disease. All right, that was the highlight, and I see you guys tomorrow for final conclusions.